with pandas, you get quite a lot of tools to inspect your data and get a feeling for your data. We had covered some of them already in the last lecture. Like reading in the data frame is obviously the first thing you need to do. And we are using CSV files, so you're reading in with read CSV. You can also read in Excel files or other file formats. Look at the pandas documentation if you're interested in that. Then the first thing I usually look is at the shape, at the data types, and do some summary statistics. So we've covered that in the last lecture. One important area is checking whether there's missing values, which can screw up your analysis. So we will look at the is-null method in detail and also getting a feeling for the categorical data like cell lines, lineage, how many of them, what are the types you're dealing with. And that will then give you an idea how to group your data and how to analyze it and compare things. Let's have a look at the notebook to go through this. So as usual, we import our data with pandas SPD. Then we load the data. I've done that already to save some time. Then you do your head and tail, for example, to have a look. Looking at the shape. So if we run this as usual, we have 89 cell lines, so similar to the data set we used in the last lecture, and 17,130 genes. It's quite good to get a feeling for what the data types are, how many columns are numerical and how many columns are non-numerical. And here the first columns are all metadata, so they're called objects, but they're usually string data. Then to get statistical analysis, we can use the describe method, which again gives us this very long data frame. So if we use the .t transform, this moves data around. So now the genes are in the row, in each row is a gene, and we get the count, the mean, the standard deviation. So this is quite a nice way to look at this data quickly. So now let's talk a little bit about missing values. They're often annotated as none or not a number values. And it's quite important to figure out whether they could be a problem in your analysis. If there's a lot of missing values, you might not want to go further with your data, for example. If there's a few, there's different tricks to deal with them. In this lecture, we're just going to look at the is-null method that gives you an idea of how many of those none you have. And then in the next lecture, we'll see how to deal with none values, because for statistical analysis, this can become quite important. So the is-null method gives you an idea of how many none values there are. And if you do a single sum, you will get a new data frame, and each column will give you how many missing values are in each column. So this looks like we don't have any, but we don't see all the columns because there's obviously too many columns. So a simple trick to get the actual total number, and this is the first thing I would look at, is to do dot sum and then on dot sum again. So we're summing now again over this one column. And here, when we do this, the total missing value is actually just one. So we have only one single missing value. And so that's not bad for, but to find out in which column this missing value is, we use this code here where we do a Boolean to check which of the columns does have missing counts. And that tells us that the missing value is in the oncotree code, which is the metadata, so we don't need to worry too much about that. I will talk a lot more about this in the next lecture because this is quite important. So now, finally, let's look at the categorical values. The first thing I usually do is check what are the columns, because that already tells me quite a lot. So if we run this, the df.columns attribute here, we are getting each of the column names. And again, this is too long, so we don't get all of the columns printed out. But here, this is quite useful because we can see all of the metadata and then the first gene, and then you get 17,000 different gene names here lined up. But this gives us an idea of, okay, these are the categorical data here. And then there is quite a useful technique to just have a look at what is the number of this. So how many different model IDs, cell line names, strip cell line names do we have? And the the method here is n unique. So if we run this, and here I'm just running it on the first 10, so I can see the actual data for each of these columns, so until the first gene. So you see that these are all individuals, so each we have 89 model IDs and 89 cell lines, so each row has a different value, but we have only 2 oncotree lineages, so that's interesting. So there's 2 groups, and then we have 6 primary disease, and we have 15 subtypes. So this tells me these are the interesting groups that I might want to compare. So then there's a really useful method, which is the unique method, which gives me the actual values. So let's say, for example, I want to see what are the 2 major groups that we have here in the lineage. So if I run this, this will give me so it's breast and myeloid. This will give me an array, and I can iterate over that or do different things with it. So this gives me now, so okay, I have here breast cancer and myeloid cancer, and then I can separate those out and build separate data frames for those to look at them further, for example. Or we can have a look at the primary disease, where we have 6 different values. So again, with unique, we get the different categorical data here printed out. So we've got AML, we've got invasive breast cancer, and so on. These are a bit more complicated cells that print you out a lot more information about these individual values. So have a look at this by yourself, what this does. And in the end here, we can print out a summary of the data quality, for example, that gives us a good information. So it's quite good to build this have a function that gives you this data, and then you can run this quickly across your data frames, and you get a quick initial idea of what these data look like and whether there's some problems with the data or not.